All right, guys, here is your biology update for Monday, August 31st. This is what we discussed in class if you happen to miss class Monday or Tuesday, or if you are a full remote student. If you're a full remote student, if you have extra questions, please make sure to join in on the Google Meet that is happening after school Monday or Tuesday or on Wednesday. So you'll notice on the classroom website, I've broken up the spreadsheet into A days and B days. This is kind of for my own sanity, and also I think it's going to help you guys out a little bit. And remote students, if you're fully remote, I would like you to follow the B day schedule, because that is going to just make sure that I know you're on the same page as somebody else. If you are a temporary remote, you are just going to follow the day that you have. And I will be posting videos as much as I can, updating you on what happened in class. So today what we discussed was making sure we had our lab setup done from last week. If you did not, then please watch that video and get it set up. It is very simple. This is actually the lab setup that I did as an example. And then you'll click on the lab worksheet. The lab worksheet is going to link you to this bread mold lab report. What's nice about what Google Classroom has done is when you click it open now, it's actually going to automatically assign you your own copy, and that copy is going to be included with your name in the title already. So I'm going to run you through a few different things that we talked about in class today, and I need you to update it on your own. The next time you need to access this, if you come back to this page, it should pull up your saved copy of it. And if for some reason the information is not there, please search your drive and then drag and drop this file into a biology folder so that you have it all organized and straight. So when you come into the lab, make sure you put your name, the start date that you put together your lab, and then also your class period and the day you would attend. If you're remote, please just put your class period so that I can make sure I put it in the gradebook under the right hour. For lab background, I'm actually going to discuss that with you really quickly, and I will speak here about what the background is about, and then I want you to type in 3 to 4 sentences on your own, kind of summarizing what I'm saying. So the background for this lab and any lab that we do is going to involve why we're doing the lab, what importance it has, and how we can relate it to what we are learning. So for this lab, we are essentially taking bread and we're growing a combination of mold and bacteria. Now, we can't really separate the mold from the bacteria as we're just looking at it. It's just the nature of how we're going to be doing this at home, but it's going to give us a really great idea of what method of sanitizing or cleaning is going to keep the bacteria and or mold at bay the longest, meaning that is the safest method for us to use as well to make sure that we are keeping germs and bacteria away from ourselves. And the reason that this is going to relate to biology is that in biology we're talking about living things and how they interact. And believe it or not, bacteria is one of the living items that we're going to be discussing. Mold, we'll discuss if that is a living thing later on, but how that bacteria interacts not only with us, but with the different ways that we try to, quote unquote, remove or kill the bacteria from any surfaces we have. Now, there are such things as good molds and good bacteria. In fact, we discussed in class how cheese is actually a mold and we eat it and it's delicious and it's fine for you. But that is in a very controlled environment and it's a specific type. Not all mold is good for you. And just like that, all bacteria can't be lumped into a bad category. There's good bacteria and bad bacteria. In fact, your mouth is made up of thousands and thousands of bacteria cells, and they are doing great things for you. And then there's also the bad bacteria that can make you sick or weaken your immune system. So we're going to try to use this information with the current day and kind of era that we're in, where we're very health conscious and very sanitization conscious and trying to keep ourselves safe from different types of bacteria or viruses that are traveling around. And we are using this as an experiment to test to see what we feel the safest with using in our own lives. So you're going to take key ideas from that. Please do not type word for word what I just said, and put that into 3 to 4 sentences for your lab background. Then for the variables, this is pre-typed from this last hour, so I'm just going to leave it there, and you can feel free to copy down what I have. It is going to depend on what version of the lab setup you did. So for me, you would have seen in the lab video, everyone is going to have a control. And then I chose to rub my hands on this piece of bread without having washed them. And then I used sanitizer. If I would have brought another piece of bread, I would have just done also hand washing for one piece, but I only brought 3 to school that day. So our independent variable is what the scientist changes or controls in an experiment. So it's the thing that we're testing between, the thing that we want to know what the result is going to be. So in this lab, we are doing the different method of cleaning or sanitizing. And for me, I used my hands. You may have used a different surface. Please, again, no bathrooms. If you did use anything in a bathroom, I would like for you to start the lab over because we don't want to grow colonies of fecal matter. And then I listed out the different ways that I controlled that. So my control variable is not an independent variable. This is just like a base that I can say, okay, this is how fast bread molds on its own. And then I'm going to compare it to what I see in my no wash and my hand sanitizer. So these are the 2 different things that I controlled in the experiment. Then your dependent variable is what you measure. And what we're measuring is going to be the amount of mold and bacteria growth growing on the bread. Now, the reason I lump mold and bacteria together is because bread is going to mold naturally. If we had our agar plates and we could do little smears in class and we could, grow the cultures naturally, we could probably separate that out a little bit better. We'd be able to visibly see a difference. But for right now, everything we see when we grow these is going to be a combination of mold and bacteria. And then I'm going to tell you the way that we're going to do that is on a scale from 1 to 10. We're not going to give these things a scale, or we're not going to rate them until we're all done, which is why we're taking pictures, but we're going to use a 1 to 10 scale in the end. And then my control variables are all the things that stay the same. So if I'm trying to test the different methods of cleaning, everything else has to stay the same. Otherwise, I have no clue what actually affected the growth on my bread. So a few things that we listed out where you want to use the same type of bread and the same loaf of bread. If you use bread from 2 different loaves, they might have been made different days, so one might mold faster than another. And if you use like banana bread, which has a high moisture content because it's got bananas in it, versus sourdough bread, and you try to use them in the same experiment, it's not going to work out so well. Also, the location that you keep your bread in is going to be very important because if you had one next to a window, I can tell you that sunlight is going to help it grow faster. And if you put one in your closet, not so much. So you want all 3 or all 4 of your pieces of bread to be in the same location. You want to use the same method of sealing. We call it just the baggie in this one. So if I use, maybe I use plastic wrap on one of these, and then these bags on another, I can't guarantee that the plastic wrap seals the same as the Ziploc baggies do. So I might be getting an extra moisture, extra bacteria that's coming in to help my bread mold faster. And then the last thing that we came up with is that it was all set up the exact same day. So we created, we created all of these bags on the same exact day. If I would have done my control on one day and I sealed it up in this airtight bag and these pieces of bread were still just in the bread bag, that's not sealed super well. So I wouldn't know what this was exposed to that was different from this piece. So you want to make sure that it's all set up on the exact same day. Then you're going to put in your location of your samples. So for mine, I put them on my front whiteboard in my classroom next to a window. And I said how much sunlight it gets. So it gets approximately 8 hours of sunlight a day. I don't need an exact number. If you are putting yours in a closet on a top shelf because that's the only place your siblings or your animals can't get to them, then that's fine. You just say 0 hours of sunlight. And we want to know that because in the end, we're going to look at a couple of different examples and see if the light played a difference in it. Then our hypothesis is always going to be an if-then statement. And your if part is setting up a summary of what the experiment is covering. So we're saying, if bread is contaminated to surfaces cleaned with different methods, which is my hand on the no wash and my hand with sanitizer, then, and here's where you pick a winner, so then this one will grow the most mold and bacteria. So maybe you think the sanitizer is going to grow the most mold. You would say then the sanitizer bread would grow the most mold. Or you can say the sanitizer bread will grow the least mold. But you're going to pick which one you think is going to either be the most effective one way or another, so that we can take that information and compare it at the very end. Now remember, there is no right or wrong as a hypothesis. It's just an educated guess. So we're just trying our best to see what we think is going to happen so that we can learn from it, because that's the whole point of science, is learning and understanding. Then you come down to your data collection. So you're going to insert photos. You can either do 3 or 4 different photos, so one of each sample. So I could take a picture of just my control and insert it, just my no wash, and then just my sanitizer. Or because they're all up here together, I took one picture of all 3 of them. You do want it to be pretty clear so that you can see how much mold later on when we go back to give them a scale. This was taken with the camera on my computer. So what you would do if you don't have another camera, the easiest way to do this is actually to have the Docs app on your phone so that you can insert it from your phone images right there. It's going to give you the clearest picture too. But you would come here to insert image if you're going to use your computer and say camera. And then you would take the picture and you would highlight over here and say insert. I'm not going to insert a picture of myself, but there you go. The other thing that we discussed that will be super useful is making sure you put a date up here of when that day 1 photo was taken. So there might be days where we forget or where we need to go double check what day we're supposed to take the picture. And by having that date there, it's going to be really helpful to make sure that we know when we need to update for our every other day. We're going to try to continue this for 11 days. We're assuming that we're going to have some pretty significant mold growth by then. We might extend it out later if in class we're discussing that it's just not happening very fast. And then we'll create our table later on, and then also our conclusion. So for right now, I just need you to get all of this top portion updated. And then you'll see in your weekly work spreadsheet, I'm reminding you when you need to have your second photo and your next photo and your next photo taken. So as long as you're going to follow the spreadsheet, you should be all set. All right, if you have any questions, please feel free to jump on a Google Meet, or I'll see you next time.